All right, here's chapter 3, part B, and this is mostly about active transport, which goes against the concentration gradient, and self-signaling, how cells talk to each other. Okay, so active transport is the one that uses ATP, goes high to low, and then vesicular transport is going to be endocytosis and exocytosis, when pieces of the membrane pinch off to let things in and out of your cells. So both of these use ATP, which is energy, right? Both of them deal with large molecules that are not, maybe they're not lipid soluble, or they go low to high instead of high to low on the concentration gradient. So they go against. Okay, so active transport has carrier proteins, just like facilitated diffusion, and these carrier proteins carry the substances high or low to high inside the cell. There's two kinds. There's antiporters. Those are the ones that when one thing is coming in, another thing is going out. And then symporters, when two different things come in the same direction. So the names kind of make sense, but that's what the two types are. Okay. There's also primary active transport and secondary active transport. Primary is when ATP is directly being used to make like the transport happen. And then secondary is when it's indirectly being used somewhere else, and then that causes the transport to happen. Okay, so primary active transport, the most common one that we talk about is the sodium-potassium pumps. And I think like a large percentage of your ATP is used for these pumps. This is how your nerves work, your muscles work, things like that. Super common pumps in the cell. But we also use them for hydrogen pumps. So when we do cell respiration, when we get energy from glucose, we use hydrogen, and then calcium is also another one that's transported through primary active transport. So the sodium-potassium pump, most studied, super common, really, really important one to know. It's called the sodium-potassium ATPase pump. Remember, if it ends with A's, that's an enzyme. This pump pumps Na out and K plus in, so this is an antiporter. So the Na and the K go opposite directions. There's also, remember the leaky channels in the last part? There's leaky channels that are going to return the sodium and potassium back to normal. This is like after a nerve impulse goes through. And then the pumps are the antiporters that are going to pump it back against the concentration gradient. So used in muscles and nerves. Okay, so here is a picture of it, and it's kind of, I took out some of the slides so there weren't so many, but the other slides just show each little frame. Here you can see, I'm pretty sure the yellow is sodium, and for every 3 sodiums that come in, 2 potassiums are going to go back out. So let me make sure I'm seeing this right. Oh, no, I'm sorry, I'm seeing it wrong. So in this picture, let me take my little laser. In this picture here, 3 sodiums are being attached here, and the little carrier is open going downward. Then it's going to open up to the outside. 3 sodiums are going to leave, and then 2 potassiums are going to come in and be brought into the cell. So it wouldn't make any sense if it took out 3 sodiums and brought in 3 potassiums, because it's trying to change the charge. And they both have the same charge. Both of them have a positive 1 charge. So what it has to do is take out 3 sodiums and then 2 potassiums, and then that's actually going to change the overall charge in the cell. So that's how an ATPase pump works. The part I didn't mention was that ATP is used. So you can see ATP here is like sticking the key in the ignition, and that's what actually powers the carrier protein. It drops off a phosphate here, that's the little purple guy, and then that's going to cause the pump to open and close. And it's going low to high against the concentration gradient using energy. Okay. Oh my gosh. Secondary active transport is the one that uses it uses ATP, but not directly. So the ATP isn't directly running the pump. It's like an indirect thing. So let's see, low sodium concentration that is maintained inside the cell strengthens sodium's drive to want to enter the cell. So sodium can drag other molecules with it, and that would be the symporter if they're coming in with it. So that would be like sugars, amino acids, they come along while the sodium is doing its primary active transport process. So here's sodium doing its normal thing. I'm always backwards. It's going out. But in the process, these other little glucose molecules also sneak out or in, or whichever direction. They're kind of sneaking with the sodium. And so that would be the secondary active transport. The glucose is doing the secondary. Okay, vesicular transport is when you have really, really big molecules that can't go through channels. They're too large. And so you have this special like pinching off process that's going to allow big stuff to come in. And it usually requires ATP. So you have endocytosis and exocytosis. Endocytosis is bringing the stuff in. So endo means in. There's 3 types: phagocytosis, pinocytosis, and receptor-mediated endocytosis. Phagocytosis is big stuff. Pinocytosis is small liquids. It's kind of like the cell drinking little drops. And then receptor-mediated endocytosis is where you have little receptors on the cell. And then when something binds with those receptors, that causes it to like pinch in and bring it into the cell. And then exocytosis is just the opposite of endocytosis, and it's taking it out of the cell, like taking out the trash. And then you have also transcytosis and vesicular trafficking. So those are like other versions. Okay, endocytosis. What happens is a piece of the membrane is going to engulf the substance and then grab it and kind of like amoeba-like fashion, bring it in and pinch off, and then it's inside the cell. So it's going to make a little vesicle. The substance, whatever it's bringing in, is going to be in a little vesicle. And then if it involves receptors, it's the receptor-mediated one. Some pathogens can get into the cell like this, kind of like a Trojan horse. This happens a lot with white blood cells. They engulf bacteria to protect us from bacterial infection, and then they eat the bacteria. So here's what it looks like. Something needs to come in, but it's too big to get through the membrane. And so it gets grabbed by a piece of membrane, kind of bubbles off, pinches off, and then you have the stuff with the membrane around it. And then these little proteins are sticking out. They're going to fall off and be dissolved. And then the cell can use the stuff. It can either like right here, it's fusing with a lysosome, and that's the digester organelle. So it's going to digest it and maybe get energy out of it. Or it can ship it out of the cell, or it can use it. It's somehow bringing the stuff in. Okay, so phagocytosis means cell eating. Phage, phago means eat. There's the vesicle that it makes, that it's like the little sac that it's in is a phagosome. And it looks like a little amoeba situation where it's like kind of alien-like and grabbing the stuff on the outside. And this is what red blood cells do. What did I say? This is what white blood cells do. Sorry. So here's phagocytosis. It's grabbing this big thing. Sometimes there's receptors that connect to it, and then that causes the cell to know, like, hey, let's grab this. And it makes these little pseudopods, which are called false feet. And so that kind of grabs the substance and brings it into a little sac. And then this is called, the sac here is the phagosome. Okay, pinocytosis is known as cell drinking, and it's just little bits of liquid, but they're too big to go through the membrane. And one thing to note is that, that sac that's around let me go here. This sac, the vesicle that's around the stuff, is going to go back and become part of the membrane again, probably through exocytosis. So that way the membrane stays the same size. It's not like this keeps pinching off until the membrane's all gone. So it's going to grab a little bit of liquid, gulp some water, bring it in, and then you have it in a vesicle, and then it can be used. Okay, and then receptor-mediated, we've kind of seen that, but you have these little receptors that are going to be triggered, and then it's going to cause the endocytosis to take place. So it happens with enzymes, low-density lipoproteins, iron, insulin, and viruses like diphtheria, cholera, and toxins. So it does it for good things, but then these other bad things have also kind of hijacked the system. Let's see, caveolae have smaller pits and different protein coat from clathrin. So they use a slightly different process. So here's what it looks like. You can see these little receptors receive the message, and then that's going to cause a vesicle to form. And then they might be digested by a lysosome. Okay, exocytosis is the opposite of endocytosis, and it's going to send something out. It's going to eject something in a little vesicle and just shoot it out of the cell. So things like hormones, neurotransmitters, mucus, cellular waste, those are all ejected out of the cell by exocytosis. So here is your example, and you have these little proteins that connect, like the vesicle can connect with the membrane and connect there. See, they kind of twirl together, and then that's going to allow the membrane to fuse with the vesicle because they're made out of the same thing. They're all made out of phospholipids, and then it's going to release the substance. So there's a picture of exocytosis actually happening. Okay, membrane potential. This is important in nerve cells. This is how your nervous system, your neurons work. When there's no nerve impulse going through them, they're in a resting membrane potential, and they're negative. They have a negative charge inside the cells. And you guys haven't really seen what a neuron looks like yet, but it's like a long, skinny cell, and then it has a head. Usually it can have different shapes. That's usually how it's shown. And when the nerve impulse goes through the cell, the charge inside the cell goes up, and it goes up to positive when it's being triggered, and then it'll drop back down to normal. So resting membrane potential is when no charge is going through it. So for this to happen, you've got the leaky channels. They play a role, and they're the ones that kind of get the membrane potential back to normal. So the potassium inside the cell is leaking out of the cell, and it's going passive transport, facilitated diffusion, high to low. And so that's going to make the inside of the cell more negative because the K plus is moving out, and then once the charge is going through it, the K plus is going to be pulled back in, and that's going to make the inside more positive. Okay, sodium also plays a role here. So if sodium moves into the cell, it moves in because the inside of the cell is negative, so it can also cause the charge to change. Chlorine is there, but it's not really playing a role because it's the same inside and out of the cell. Okay, so here it's showing the leaky channels. Potassium is leaving and potassium is coming back in. And at this point, the charge inside is overall negative. So you can see the little negatives here and the little positives here, and this is when your neuron is at rest and there's no nerve impulse. There's no action potential going through it. Okay, so resting membrane potential stays the same because of the sodium-potassium pump, because it's putting out 3 sodiums and bringing back 2 potassiums, and that makes it uneven, and it makes it negative overall because you're only bringing in 2 potassiums, even though they both have the same charge. When you get a nerve impulse, then that's going to change. Okay, and you'll learn more about that later when you study the nervous system. All right, so there's a couple ways that cells talk to each other, because they have to be able to communicate in your huge body of trillions of cells, long distance, like it's really far for them. So they have a couple mechanisms. One is that they use the glycocalyx, which are those little sugars, those carbs that stick out on the cells. They have cell adhesion molecules called CAMs, and then they have plasma membrane receptors. You kind of probably are, like we talked about that before. Okay, so CAMs are little things that stick out. They allow the cells to anchor to extracellular matrix, to each other. They can help them slide past one another. They can bring white blood cells to injured or infected areas, help facilitate tight junctions, or they can cause tight junctions to form or degrade, and then they can also send signals, and the signals tell the cells what to do, because otherwise they wouldn't be able to maintain homeostasis and talk to each other. So it's pretty complicated, actually, how they communicate with each other. So there's contact signaling. That's when they're actually touching each other. They're touching their surface membrane receptors. They're like, hey, who are you? And they're able to communicate that way. That's when they're right next to each other. But when they're long distance or even like nearby, they're going to use chemical signaling. And they use what we call receptors. We've used that word. And then ligands. I have said that word before. Those are the little chemical messengers. Ligands bind to receptors, and then that tells the cell to do something. And it, like, maybe it tells the cell to grow or make a certain hormone or something like that. So yeah, it can cause it to make an enzyme. It can cause it to open chemically gated ion channels. Some ligands, that, like a ligand you might have heard of is like dopamine. That's a neurotransmitter, and it talks to your cells and tells it, like, kind of gives you the response that you're happy, right? So that's what neurotransmitters do. The ligands are specific to certain receptors. They have to connect to the right receptors. And some ligands can connect to receptors on different cells, and then they're going to cause a different message. So yeah, they can do things like open ion gates, cause a nerve impulse to go through, things like that. There's also these G protein linked receptors. These are super common. They're in the cell, but they're like right near they hang out right near the membrane, and they like transmit the message to the inside of the cell. So they kind of start the cascade or continue the cascade and the ripple effect. And we will also see, this is called CAMP, cyclic AMP, or calcium. Those act as secondary messengers. They also pass the message along. So this is showing what that looks like. Right here in the pink is the ligand. It binds to the receptor. And then right here's your G protein that hangs out right on by the membrane. It gets changed, and then that's going to cause another response to happen. So it's kind of like I feel like it's like passing the ball in basketball, where this one gets the ball and then pass to that one, pass to that one, pass to that one, and then the second messengers are here, and then eventually they're going to get to the nucleus where they pass on the message, or somewhere else in the cell, and they tell the cell what to do, and that's the basket. That's the point. But these are all the players that are passing the ball like in a relay. So that's how cells signal to each other to tell each other what to do. And that's it.